think we put them in some Google Drive, which apparently was not really appreciated by some of us, including me.
So, I mean, I was told we were allowed to one slide, so my slide is very busy. Can we project my slide? And I'm going to time myself. Okay, well, while we wait to project my slide. So the Immune Bio Network now has over 260 members. Actually, this is probably an underestimate because I think we had 20 new requests lately. So sorry for the very busy slide. It's mostly for me and my notes. And so we had a lot of we tried to have a lot of community engagement efforts over, I would say, the last 8 months. We had 4 annotation meetings. This meant mostly hearing from different groups how they're approaching their immune cell annotations across their own tissue. We had 6 brainstorming sessions across different time zones. So every session is hosted 10 p.m. to midnight on North American time zone so that we can accommodate Asia Pacific, and then the next day for North America and Europe. And what we've been doing is we have a live Google Doc, as well as a live PowerPoint, where we've been gathering everyone's feedback in order to try to build our roadmap. That's how we achieve it, using the roadmap template. Currently, we have a draft, and we're trying to, it's a live document on a Google Doc, and trying to gather feedback. In terms of the data sets, it is a little challenging, at least for the Immune Bio Network, to gather their data sets because immune cells are across all tissues. And so what we've agreed across our BioNetwork contributors is that we should think of it about 3 different tiers for our data sets. So first, there's tissues that only us are gathering. So these immune tissues would be blood, that is peripheral vulvar cells, bone marrow, cord blood, tonsil, thymus, spleen, and lymph nodes, which is across the body. Tier 2, and I'll get back to this, Tier 2 would be mucosal barrier tissues that are more enriched for immune cells, so that would be the GI tract, the lung, and the skin, and Tier 3 would be all other tissues. And I'll get back to the different data sets as I go through challenges. In terms of modalities, the reality of it is that we have to go with what we have at this time, and we mostly have single-cell RNA sequencing. We don't really have open chromatin. We have site-seq for some data sets. We have repertoire for some data sets. We have very limited spatial data to a point that I don't know that it's going to be that helpful for version 1. And we simply don't have multimodal data at this point. Data gaps. We reason that putting together our data sets will help us define these data gaps, but in reality, we know that we're missing cells. For example, we're missing granulocytes, which is a huge component of immune cells. This is just our data gap. We have modality gaps. Sampling diversity, we're trying to work with you heard Shaham before, we're trying to work with their BioNetwork to kind of integrate all of our data sets together. And we know we have data gaps across donors. So I'm going to go across challenges that we're encountering as a BioNetwork, which also has some impact on the computational needs. The first one is actually the data set identification. I didn't think this would be so challenging, but it's surprisingly challenging. So we use a survey that Aviv circulates every quarter. We circulate our own survey. We went through all the grantees from the CZI networks, from the hub map, reached out to people, trying to do literature review to gather these data sets, and made several calls through email, Zoom, and Slack. And again, if any of you are listening, I don't know, this is not virtual, it's going to be recorded. Please tell us if you're working on a data set. It's okay if it's not ready. We'll know for version 2. So that is a huge challenge at this time still. So before I go back to challenge number 2, I can go in terms of what we have right now that we have identified. We have, I would say 15 to 20, we have probably 20 million cells, but most of them are blood. Nothing wrong with that. We can learn a ton from the blood. We have about 1.5 million cells of bone marrow. There's probably more that we're hoping to identify in the coming next few weeks. 1 million cells of cord blood. We've identified 5 data sets of tonsils. I'm pretty sure there's more than 1 data set of thymus, but right now there's only 1. I'm pretty sure there's more than 1 spleen. So if you guys have spleen, do let us know. But currently, this is not 20K, it should be 200K. And then there was Cecilia with us, recently published a beautiful cross-tissue data sets along with other data sets. So lymph nodes, it looks like we mostly have one-site providers. So one of the challenge here is that while we have a lot of data providers for blood, we mostly have specific sites for other tissue. And so the other challenge quickly, so immune cells are in all tissues, so we need computational groups. So we have different computational groups that are involved in analyzing immune cells. They don't necessarily analyze immune cells specifically, but everyone is somehow analyzing immune cells, so we need coordinated effort across bionetwork and computational groups to extract these immune cells and build annotation strategies. I have a hard time developing common coordinating frameworks, but the reality of it is every tissues are developing their own frameworks for immune cells, so we need to coordinate there. We need to think about integration of data set by tiers, because you need to integrate first all the different immune tissues separately, and then bring them together, right? So first per compartment, so that's another challenge. We have the challenge of continuum. Cell states are not binary, and we're treating them like binary. And so that's a big challenge. And we have a challenge of scalability when it comes to, don't have slides anymore, but when it comes to 20 million cells, that's a challenge. So 20 million cells, we don't want to downsample necessarily because we are trying to figure out all rare immune cells and cell spectrum. And the last challenge is related to annotation, which is going to evolve and we're going to get wrong at the first time, but trying to figure out how to come together. And you heard about the Immune Cell Atlas, Immune Cell Annotation Jamboree we're trying to organize together with the Genetic Diversity Work Group. So I'm going to stop now because I'm at 5 minutes flush. Can imagine my slide. And so to sum up our challenges, we can still proceed with data set integration because we have a lot of cells. I think some other tissues will be proceeding and coordinating with some of these bionetworks to work with them to extract their resident immune cells, and work together on this is very feasible upfront. And then just the continuum problem, the scalability problem, the annotation problem. I will stop here, Aviv, because I could carry on, but I'm going to stop here.
Do we know where?
We have the slides. Are they ready to go up?
If not, we could maybe discuss you
Yes.
first and then
Next
do
slide.
the others. Let's do you first.
Okay.
We'll adapt. We're
Yeah, of
flexible.
course, of course.
So we have a series of questions. They're going to repeat themselves for I'm not sure. They might show up or not. If not, I'll read. I have actually, an old-fashioned piece of paper, which is quite convenient all of a sudden. I've gone very paperless in my life, so this is an unusual experience. But let's start by Chloe by asking a generalized question, which is whether there's sufficient, Chloe and the audience, sufficient genetic, gender, geographic, and age diversity in the data sets that you have presented today, and are the metadata sufficient for diversity analysis? And if not, which is almost invariably going to be the answer, actually, probably in blood,
So I was going to say
it's the one where you have a potential to answer yes. But if not, what steps can be taken to increase it?
So that's I forgot to mention this when presenting. So yeah, for blood, so we even involve the genetic biodiversity group, genetic diversity bionetwork group in this conversation. So blood is probably the only one where we have captured to some extent this. And we had this whole discussion, which I forgot to mention, disease versus no disease, which everyone is having as a discussion, but it's in the case of immune cells, it's something to consider because if you really want to map the full spectrum of immune cells, you need to consider some form of perturbation. If we include COVID, for example, it's to your point of diversity, you start getting cohorts that are across age range, from pediatrics to 99 years old, across different backgrounds.
Probably the size of everything else that's been done because so much was done for COVID.
Correct. So we have an opportunity in the context of COVID with blood to start answering these questions. You can start downsampling and ask how many donors would I need across ancestry and age group to define all existing cell states. And that could help inform the analysis of other tissues and so forth. So there's an opportunity for blood. For all other tissues, even in one ancestry background, we don't have enough. Because in most cases, we have like 10 donors, very specific ancestry background. So I would say the only place we have it is really blood.
So there's an opportunity with diversity, with ancestral diversity in blood, and also in blood there's likely an age opportunity now.
Correct.
Specifically because of COVID, or in particular?
In particular because of COVID.
Other thoughts on that, on diversity in the context of immunity from people in the audience?
Great if the lessons learned could be communicated widely, because it probably will be the one.
Yeah, we need to start using
to
that.
do best.
AD person. How do you turn the mic?
Usually there's this
we made it a task as part of our jamboree actually to figure out the minimum number of donors to define.
Does this one work? Great. So I'll move this one here, and you can use that, and we're good. Now we're wired up as well. Okay, great. This will be the kidney when we get to it. The kidney will come up soon.
Chloe, you has to turn it on in the back.
Yeah. Okay.
They need the mics on.
So now let's turn to Atlas integration in the immune system. Our first question is, are all the relevant data identified or listed for integration? Or you might have to use this one.
Yeah, I mean, I think that goes with the problem of asking everyone to give their data sets along with the metadata. I think we're getting closer because we're nagging a lot of people to gather this information. I think the other thing that's coming out very clearly that we'll need to implement for version 2 is that while we aspire to have standardized metadata for version 1, we didn't really achieve this. But there's a lot of lesson learned here, so that as we improve all our gaps, we can actually try to have a minimum standardized metadata, and that will really help improve. But at the very least, we know the methods that were used to generate the data, the chemistry. Actually, I should say that the metadata for data generation is pretty good from what I've seen. And so in terms of thinking about the technical artifacts, this is actually pretty decent. I think where we're missing is actually metadata about the demographics. Yeah. It varies tremendously depending on the IRBs and the background and how samples were collected. But you know age range, ancestry background, gender. Well, you can figure out gender.
Well, that you can figure out sex at birth.
You're right. Sorry, sex at birth. Just correct. But sex at birth.
Genetic side you can
Sorry. Genetic size of gender. Thank you, Aviv. Yeah. And so I think one of the other big, maybe that's one of your other questions. I'll let you ask the questions.
You let me ask the questions. Others in the audience that want to maybe comment on this question of identifying the data that's out there? Oh, I'm sorry. And I would say in this context also, how important it is in people's minds to integrate data that's published, where you can always obtain it from external sources as a result, versus obtaining the data that exists today, which includes unpublished data. So in the lung example that we saw, there's also unpublished data. It's not all data that has already appeared as its own individual paper. Where do you feel the community is on that, and the community of this room?
I would say, I don't know if this is on, but I would say that for the Immune Bio Network, we definitely have a few providers, I would say Microsoft including, that are provided unpublished data. We're going to try to coordinate so that the postdocs have their paper at the same time as we integrate.
Yeah.
I think there's tremendous value, so we remain cutting edge, and I think everybody can win through the process, and we're doing it to lead by example. I think that's important.
Great.
The reason I'm going to encourage everyone to do it, I don't think we lose anything. We're going to make sure that everybody gets their own paper. We're just going to standardize carefully. The reason we are trying to query the published data set is that there's so many gaps in the tissues, so we're trying to see what's out there. The problem is that a lot of it is disease. That's a lymph nodes of disease, and that's not what we're looking for version 1.
Yeah.
So, yeah, it's important.
One thing that occurs to me is that as we have been talking about publication bundles, is that could be a way of doing a bundle. That the kind of the front paper is the integration, and then there's the series of the individual papers that are also doing the deep dive into the different biologies and questions that people might be interested in.
And just to add to this, I think anyways we need to analyze it separately so we understand what the
Yeah.
data looks like.
So in terms of the granulocytes that you were mentioning, as in, they seem to be the most important, or in terms of abundance at least, but almost nothing is represented. Is there a plan for that? Because we're working with some new technologies from Hive, honeycomb technologies, and another one, a couple of technologies which claim they can do granulocytes, and they have some pilot data which looks promising. So if we were to do that, what would be the plan? How would you integrate? Because you already have 20 million. It's not, so you cannot go back to collect them. So what would be the plan?
I think it is so. I'm glad to hear you're interested by this effort. I almost feel like granulocytes needs its own dedicated effort because they need to be handled very differently. They need to be handled fresh. It doesn't do well with cryopreservation. And you're
Yeah.
Abundant cell type. So when it came to, to our brain networks, they find the gaps were like, yeah, we're missing granulocytes. So it was like an obvious gap.
Chloe, can you open your mic up more? We're getting some audience, virtual audience feedback.
Oh, that they can hear us?
Yeah,
Okay.
just Chloe though.
And we will also start moving on in the questions, or we will never get there to the other organs, which we definitely are determined to get to. Organs and systems for the adipose. Is the data available in raw as well as similarly processed format?
No. Oh.
Okay,
No, this
so
is
basically
definitely
all my
working.
questions are going to first be answered with no. It sounds like talking to, your...
Sorry, I don't mean to be I think these are all part of the
No,
challenge.
it's the state of the...
It's the state of the field, but
Yes.
I think what was helpful in our good practice breakout was learning from the lung, who kind of established a standardized pipeline that they distribute across different groups. So at least you kind of have the cell, let's say you use 10x the cell ranger version, the same reference. It's all very feasible if we kind of get a bit more organized.
So my next question, first I'll let the audience vote on the answer, and then we'll see whether they predicted you correctly. Is the data QC adequate and uniformly applied? What say you? No. They say no. They're mostly polite.
I would say sort of, and that surprisingly, people have been using very similar stringent cutoff from QC, but the reality of it is that it needs to be all reprocessed to have exactly the same QC. But because everyone is using very similar filtering approaches,
It's not far off.
it's not far off.
Okay. So Chloe actually answered the next question earlier. It was, do we have metadata on samples, protocols, sample location, common coordinate framework doable? Well, not for the blood. Preparation, etc. And my summary, what I heard from you before was people collected a lot of metadata, but it's not standardized. And on some topics, especially on demographics, there are a lot of variation based on what the IRB approved.
Correct.
So that's a big lesson learned for any future data collection. Yeah.
Aviv, we have an audience question from the virtual audience.
Go for it.
In Milano, what paper are you talking about? It was 3 minutes ago. You mentioned a paper.
The paper bundles.
Somebody mentioned a paper.
So in the opening talk yesterday, we mentioned that we are exploring the possibility of additional paper bundles, I hope they can hear me, for HCA. And as Chloe described the plan for the immune network and how people are willing to share unpublished data, their approach was, we will do the integration, but each group or data generator collaboration will publish its own paper. And I said, well, that's a great opportunity for a bundle. You can have the integrated analysis being the main, it's not the main paper, but it's kind of the paper that is the most overarching, but all of the deep dives go in the individual ones. For those who saw Ed's presentation yesterday from the BRAIN Initiative, in which my lab used to participate, so I can describe my personal experiences with that, that's exactly what we did there. There was one, it was known as the flagship paper, and we're all authors on it, and all of our data came there, and all our computational methods, because it was needed for the overall. But then each of us has one or multiple papers that actually came from our work. We had both a paper on the cerebellum, where we collected data and analyzed it together with Evan McCosco, and we had a paper on tangram, which is a mapping method, which we developed in the same effort. And so both a paper like this and a paper like that, and many, many other papers, and they were all bundled up and went through all the Nature journals, and it was very nice for everyone involved.
So
That's my experience.
That answered that person's question. They were anonymous, so thank you.
To
You're
the colleague's
welcome.
question, if there's other listening to us and you have data that is not published,
That's
and you
your
haven't reached out because you're like, oh my God, I need to publish my data first, please do reach out. We're going to find ways to coordinate.
Yeah. Next one, and we need to kind of, because we have two more. How good is the current cell ontology for your system?
Well, I think we can all agree on cell lineage. We agree on like what's a T cell and a B cell. And we probably agree on what's a CD4 and a CD8.
And a macrophage.
And what's a monocyte and a macrophage and dendritic cell.
I said sort of.
But I think that the community recognized that there's a need to agree on the annotation. And
Yeah.
what has been really nice is to see everyone participating online and really wanting this annotation jamboree and really being willing to get engaged to it. So at least as a community, we are working towards, working together for a solution. And one thing that we agreed on is, thinking about, well, I guess ontology or like a hierarchy so that we can figure out at which level we start disagreeing in terms of the resolution.
Yeah.
So I have a framework for this.
So the next one you have, I believe, just answered. It was how different is individual annotation? Can we find a simple consensus, maybe hierarchical, maybe mapped to cell ontology, if not mapped already, but you kind of answered that. And so the next one is moot for you, because it says, include all cells, or how to deal with immune cells.
Yeah.
So in an organ atlas, that was a question, but of course in the immune atlas, it is less relevant. So I'll skip to the next one after that. Integrate all data sets or build an atlas using a curated subset and map the rest onto it.
That's a good question, actually, for blood, because it's so much data. Do you actually need 15, 20 million cells to figure this out? Probably not. And so figuring out perhaps an exploration and a validation cohort would be the way to go to define cell subsets. For other tissue, I think at the very least we need to release what we have, right? Even though it's going to be very incomplete, and that will not require as much integration. I think the other layer that's more complex for the immune cells is that they're part of a spectrum, but they're also a spectrum that crosses compartments. Bone marrow to blood and so forth. And so we have to figure out that layer of complexity if we're going to try to tackle it. So that's very specific to the immune system.
Great. And my last in this long list, map perturbations.
Yes.
Disease, aging, development.
So it's interesting because we have geographical disagreement in our bio-network. Geographical, because I host sessions.
I see a vigorous nod there.
I host sessions at different time zones. So actually, Asia, Pacific, Australia are all for including disease to extract, all the cell states. Whereas when I host in Europe, they are actually against it. So I actually have geographical disagreement. And so we found an in-between.
One of each?
We found an in-between agreement, which is those that get infectious disease are actually normal individuals.
Great.
They're not predisposed. And so that's a perturbation where you go back to normal. So studying infectious disease could be like an in-between as well as vaccination, for example. Okay. So that was like an in-between solution. But yeah, the geographical disagreement.
This is really cool. Anything to add from the audience?
If you have data sets.
Complete quiet in the room. What I will say is, what we're going to do next is we're basically going to do the exact same exercise, quick intro and my Q&A with, who wants to go next? Kidney. With the kidney. I can see kidney people in this audience. I wonder if they will. And after that, we will look at some general questions that I think are just the same for everyone, like what are the computational biology needs, how we organize things, and how we think about cross-tissue analysis, a little different for immune and adipose than for kidney.
Brilliant. Hi, everyone. My name is Jillian Shaw, and I'm a scientific advisor at the Broad Institute in Anna Greca's group. I newly joined this project a couple of months ago, so I do want to invite my audience members, Matthias Kretzler and the other experts in the room, to really heavily participate in this section. But I did want to provide a brief progress report on some of the data that we have assembled and acquired. So I actually wanted to start with the key lofty questions that the Kidney Human Cell Atlas set out to answer. And please do take a look at our draft roadmap because there are a lot more details in there. But what is the cellular and transcriptome of adult kidneys, and how does it change with normal human aging? What is the cellular composition and transcriptome of fetal kidneys, and how does it change during normal human kidney development? Are there differences in cellular composition and transcriptome of human kidneys in males and females that explain well-established sex differences in susceptibility to kidney disease? And what are the differences in cellular composition in kidneys in people of different ancestry, which may underlie well-established differences in susceptibility to kidney disease? So lofty questions. The way in which this is being tackled is we have a number of teams, and I think as we discussed earlier in the day, a lot of the challenges were focused on administrative details of having MTAs in place and being able to navigate some of the challenges of COVID. So I'm going to report out a few kind of astonishing data sets that we were able to upload amidst the challenges of the pandemic. So once the MTAs were established with Stanford, There were 24 living donor single-cell kidney tissue data sets that were uploaded to the HCA system by the HCA Atlas team. In addition, our Boston team has uploaded 23 single-cell RNA-seq data sets from 9 different humans and 34 spatial slide-seq data sets from 8 humans to the CZI metadata team. Sanger has uploaded 22 samples to the CZI metadata team, and of huge importance, the Michigan team has been acquiring a very unique tissue source, 20 fetal kidney tissues and protocol kidney biopsies from living donor transplant kidneys that were procured from healthy organ donors, which is really an optimal source for normal human kidney tissue. In addition, as Matthias was mentioning earlier in the day, being able to have tumor nephrectomies and to be able to biopsy actually gives an advantage in terms of diversity of what is available in terms of the OR. And all ethics committee approvals have already been obtained for all sites in this study and for these plans. Getting into the sequencing data of interest. So one challenge and one thing that I think would be hugely interesting going forward would actually be to have a working group with the immune cell group and actually look at crosstalk between our 2 data sets. This would be instrumental. In addition, I think, the vascularization to be able to get additional insights. And so I'll just briefly speak to what we've been doing on our team at Boston, but we've been really focused on high-resolution spatial transcriptomics, and our goal is really to have a high-resolution spatial reference map at various time points across the human lifespan. And so we've been using 3 different techniques. So we've been implementing a novel method, SlideSeq. I can post the science paper that this is based on, recently published, and it allows for spatial transcriptomics on 10 micrometer tissue slices. We've also been doing multi-round hybridization chain reaction, HCR, based single nucleus FISH. And we really want to get a sense of the spatial map detailing the locations of different cell populations and the interactions between neighboring cell populations. I do want to give Matthias an opportunity to discuss some of the more detailed information that's happening at Michigan, so I am going to pass the mic if you're so inclined.
Thanks a lot, and thanks for coming on to the project and coming to Vienna. Very much appreciated. No, I think it has been a real ride. Obviously, our tissues are procured by invasive procedures, so COVID had a huge impact on the tissue procurement. We were very fortunate to step in of some of the needs, both for the kidney seed network and for the standard working groups. And as mentioned, we're able to establish a sample sharing network inside the kidney seed and the standard working group. So one opportunity I think we have as kidney people is that the standard working group people are using kidney tissue to generate a lot of feasibility data, and in some instances that might help to disambiguate some of our cluster assignment and other opportunities. So I think that's one crosstalk. We should make sure we keep in touch, obviously. You can do that nicely at the Broad, as I think the chair of the standard working group lives close by. And the other piece is that with HubMap and KPMP and human cell, we are using the same tissue reference source as the standard working group does as well. So data sets generated by HubMap and by KPMP again can be leveraged against each other so that we, with the multimodal data available on these fronts, can be integrated there. And there's a bioarchive paper out, Lake et al., where actually data from human cell, HubMap and KPMP jointly are currently in review with, I think, 1.5 million neighborhoods mapped against, 480,000 single cell, single nuke data sets, which, at least for our organ system, is substantial. And yes, exactly, the immune cells are key features and key defining features of these neighborhoods. So as the immune cell working group works through tier 1 and tier 2, tier 3 kidney is knocking on the door, so as you gain insights, No, seriously, because we see, and in AMP lupus nephritis, Nia Heikkinen obviously has been working, I think, with some of you already. We see beautiful how circulating tissue resident, kidney tissue resident, and then urinary monocytes, macrophages change their phenotype, are reprogrammed by the tissue neighborhoods, and are reprogramming these neighborhoods. So that might be really an aspect where we have one organ system where you see invading and evading immune cell phenotypes and how they change their characteristics available. Yeah. Yes, we have in.
Maybe you can just repeat the question.
Yes, so the question is peripheral blood available? From some of our reference patients, there is. From the lupus nephritis patient population, for sure. And again, Nia Heikkinen, I think, is a key point person I would reach out if you're interested to see how to map that going forward. What I find particularly helpful is that you really also can see how macrophages who are emigrating tissue or recovering tissue, how they modify their phenotype as well. Okay.
Okay, I would say Argelia and Matthias ready for my questions. What is that? So I prefer you because you did the work.
Sorry, I thought something. Never mind. Yeah, you could sit side by side, and I will ask my questions. And so, is there sufficient genetic, gender, geographic, and age diversity in the data sets presented today? And I know that from, as you know, I managed to somehow make the other one disappear, but there was a lot of fetal and adult and so on. Let me try and find it. It's probably just enough. This thing is just not very accommodating.
So we have limited availability. So there is fetal tissue available, there are young adults, there's an aging population. Gender is split halfway equally. Racial representation is underrepresented. So we have...
The other presentation. This one. No, no, the one with the kid. No, it was that one. It was that one. It was just different stuff. No, not this one. This one?
Yeah, the adipose can be on
The one that said adipose? Just the first
It
slide.
is the adipose one, you just need to go
This
to
one?
slide one.
Just this one.
No, we don't want adipose
go.
tissue in kidney biopsies.
That's not for the patient. No offense, Margot. We like adipose cells, just not there. Just not there.
Actually, we regularly do get a little bit of adipose tissue, and we should talk how to.
You should all
utilize it.
hold those important thoughts, because 5, the question I have not asked yet, would be propose an actionable cross-tissue integration project for the bionetworks in your breakout. And I already heard make a couple PBMC sample every time you collect from a patient.
Yeah.
And when you run into a little bit of adipose tissue, please bring it to Margot.
Yes.
Yeah. Anyway, we'll come back to that.
So diversity, obviously, I went through
Yes.
it. So clearly, still a significant way to go. We do have significant diversity in the disease populations we are studying, similar to with the immune cell population. So that, to some extent, allows you to capture some of the underlying racial, ethnicity, and ancestry diversity. We do have an effort in place and tissue procured from a large sub-Saharan network, the HCA Africa Kidney Seed Network, where we have over 600 biopsies in storage for single nuke RNA sequencing, where we are currently looking to acquire funding to actually analyze these samples, with an effort which is obviously led by the HCA Africa CKD investigators out of Ghana and Nigeria.
Very exciting. Okay. So now I'll ask that long series of questions that's really critical for Atlas integration. Are all the relevant data identified and listed for integration for kidney?
It's still a work in progress. We do expect to have by August, actually most of the data that we intend for version 1 to be collected and uploaded, and with loftier goals for the second roadmap.
Fantastic. And is the data available in raw as well as similarly processed format?
Yes, certainly.
And is the data QC adequate and uniformly applied? I would say if one is yes, the other is probably.
Yes, and we've been very fortunate to lean on Olga Troyanskaya's team at Princeton for most of the analysis.
Metadata on samples, protocol, sample location. Is there a common coordinate framework? Preparation, etc. We're turning straight to Matthias once I said the common coordinates. It went to the kidney.
Actually, the fortunate answer there is actually yes. For most of those, they have been procured using very standardized settings under direct supervision of the procurement teams. Kidney biopsies are obtained from a specific region of the kidney, so we actually have registered some of them already in the CCF together with Kathy's group as one of the pilot studies there. So I think we are in pretty good repair there, I would say.
Very exciting. How good, quote unquote, is the current cell ontology for the system?
So the ASTC plus B table from Kathy Borner's HubMap, kidney was a guinea pig there. And we had significant effort from Todd Valerius and Sanjay Jain with myself and with my former MD PhD thesis supervisor, who is one of the nesters of renal anatomy, to actually bring which was reactivated out of his retirement, age 86,
So
and really helped us.
I have to say, a highly senior HCA member.
No, truly, I think this is one thing, actually probably for the other organ systems as well. This is really knowledge which is literally dying right now. And if we can reach out to anatomists and physiologists to bring their generational insights from the 60s and 70s and 80s. For us, it was enlightening that, particularly some in the capillary architectures and in some distal nephron segments where there was clear knowledge available from structure, which we kind of are trying to rediscover with molecular tools.
This is fantastic. This is a huge opportunity for HCA. Yeah. It's actually really inspiring. Amazing. How different is individual annotation? Can we find a simple consensus, maybe hierarchical, maybe mapped to cell ontology, maybe answered already in the previous answer, that the annotation is actually in good shape for the kidney and benefited from this legacy knowledge. But I will challenge you on the immune cells.
No, absolutely. No, I think the disclaimer there is that we certainly are recovering or reconfirming what's known about renal anatomy and physiology, but there are a tremendous amount of subtypes emerging. In the Lake et al. Paper, the group defined, I think, 96 different clusters, and that's where a lot of consensus building needs to take place. And I really love to see that some of the tools described this morning hopefully will help the community to do that. So there's a lot of work ahead of us. And certainly the immune phenotyping, I know that I'm not an immunologist, let's say it that way.
Include all cells? How to deal with immune cells, stromal cells? We know that the fat cells also stay out of the kidney.
And also the vascular beds. I think we had at a former HCA meeting. I think this is really, for us, a critical feature. In the kidney, you have a very unique anatomy that in the glomerulus, you have an arterial capillary bed under arteriolar pressures, and then you reconvene a capillary bed again with very unique oxygen tension. So there are lessons to be learned and lessons to learn from other organ systems. So we would love if there is a way to link across organ platforms in that space.
Great. Integrate all data sets or build an atlas using a curated subset and map the rest onto that?
I don't know.
Okay.
I think we really see different philosophies in play here, and I really would love to see how other networks learn from the effort there.
Go for it, absolutely. Just exactly.
Chloe Villani from the Immune Bio Network. So, to ask back about, you want to hear about other bio network, is the question more a question of sample size? Because, you may think that you haven't sampled enough the kidney to be able to do exploration versus validation. So at this point, you would rather kind of integrate everything. Is it more like sampling and number of donors that's making you hesitate?
think the point is obviously that with the epithelial cell phenotypes we are capturing, they are clearly sensitive to the different procurement protocols. And depending which protocols are used, we are seeing clear gaps. The HCA meeting in San Francisco, I think, was a good example where it became clear that each of the technology, even deployed by different groups with different dissociation protocols, had different strengths and weaknesses. And that's why probably the strategy is to be as inclusive as possible. The stringency criteria are a hot discussion topic. If you have tissue which is already partially ischemic under physiological conditions, where do you do your mitochondrial cutoff for single cell data? So this is where I think we need a fair amount of consensus building along those lines.
Map perturbations already, disease, aging, development. So aging and development were on your slide. Should I infer that they're in? How do you look at disease?
Oh, there's a lot of effort in disease in place. And full disclosure, the Kidney Precision Medicine project is kidney disease focused, but is leveraging expertise with human cell and vice versa. So we have shared protocols. And several of our manuscripts are joint manuscript between the disease community and human cell.
And do you see it when we think about integration of the atlas itself? Do you see it valuable to start mapping the disease?
Absolutely, absolutely, absolutely. Because what we see, and it's very well known, that with physiological aging, a lot of the molecular programs in the kidneys are activated. We even see structurally scarring processes in so-called healthy aging kidneys, which are very difficult to distinguish from disease processes. So that's a very strong rationale to do that.
More thoughts from our audience on kidney? Yes, two, I think. So Chloe will help with the mic. You've been enlisted to be a runner, Chloe.
I'm Karen Seil from GSK AI. So clinically, there are three broad...
Can you...
Oh, is that better? Yeah.
Yes.
I'm Karen Seil from GSK AI. Clinically, there's three broad cell types which are very important in the kidney. So the glomerulus, tubules, and the JG apparatus. How well represented are they in the atlas so far?
So right now, as we work both of tumor nephrectomies and of biopsies, the entire biopsy is processed for the reference atlas. And we do see robust representation of the glomerular cell and the particular different cell population in different technology protocols. So the single nuke data pulls more of the filter cells, which are differentiated fragile epithelial cells in, versus the single cell data is better with several of the immune cell populations, which are not as well represented as in the single nuke data. JGA, the jugular glomerular apparatus, this is a very unique place in the kidney, where the tubules who are capturing the ultrafiltrate from the glomerulus, they go all the way down to the medulla, they come back and they find the same glomerulus where they originated from, and then they signal the blood flow in the kidney. And those are per nephron only a couple of dozen cells. And we do find them, but they are in lower abundance, but they are present. Are they present enough that you can do adequate modeling of age, gender, racial differences? And the answer there is no. Yeah.
Any more? Oh, Chloe said she had a question.
Oh, I mostly like to clarify what I asked.
Sorry,
Please, please.
So there is a cell by gene instance where you can go and you can ask those questions. So have yourself fun. It's the KPMP Human Cell Atlas HubMap instance where the combined data sets, it's 480,000 cells. So it's not great, but it's good enough to start some of these answers.
And more will be coming, clearly.
I just wanted to clarify what I asked about blood, because it gets hard to figure out what's circulating from what's resident cells, especially if you're looking across compartment and urine and so on. That's why I ask about PBMCs. I don't think we need more PBMCs, but I think when you try to, for tissue specific.
It can be an easy thing to bring along for the ride.
And if If you have procurement protocols which you like to use, please share them with us. Yeah, so we clearly want to be as standardized as possible in this setting. Also, if somebody in the group is planning to do a kidney study from unaffected parts of tumor nephrectomy, and those are not normal kidneys, but they are close to normal, and we have abundant tissues, we have biopsy-type tissue cores available, and are ready to share them with whatever group wants to start a study, so that you have a reference sample available if in a future scenario you would like to integrate with human cell HubMap or KPMP data sets, which can become helpful if different data sets have limited sample sizes.
I see, sorry.
Hi, my name is Patricia. I'm from Sao Paulo, Brazil. And I'm very curious to know, since kidney is such a complex organ, when you talk about all the samples that you have already collected and analyzed, are they all looking at the same regions of the kidney? How does that work for the study design?
Sorry, you're getting your exercise. No, it's a very good question. So we try to get an answer to the overall anatomy out of nephrectomy specimens where we have ample tissue access. And actually the HubMap group has done very rigorous work to be in cortex and in medulla and go through these different regions. But obviously these are samples who have significant procurement artifacts embedded in them. For the biopsies, where we try to stay superficial, we have big blood vessels deeper in the kidney, which you don't want to biopsy. And there we have the opportunity that based to the anatomy of the kidney, you have what we call medullary rays, where elements of the medulla are actually present in the cortex. And if you sample enough, you actually will find, and you see that in the cell by gene instance, you find even in the biopsy specimens collecting duct elements, which allows you to at least get a good representation of what these cells look like. The oxygen tension probably is higher. We see that HIF activation is lower in these medullary rays than it is in the deeper cortex, in the deeper medulla. But I think for overall principle, cell state definition, it's probably sufficient.
Matthias, I probably should know the answer, but I don't. From a organ donor / post-mortem perspective, when you actually have access to a full organ, how would you look at that?
So this was very, very informative to us. Actually, it was a study, the European Renal Cell cDNA Bank, who had their second meeting in this room, 2001.
No way.
Yes, so it's really a homecoming. No, what we did already, I recognized there that we used kidneys who were not able to be transplanted due to vascular abnormalities or due to a viral infection. We tried to use them as reference material, and we saw that these were cells, organs ad extremis, meaning they had, and I think you also mentioned it earlier, that those samples have extremely high expression changes due to the cytokine storm involved. So if you are interested to really have a highly activated epithelial cell organ, then those are valuable for that, but certainly not as reference tissue. So tumor nephrectomy, unaffected parts have significant ischemia. We procure them in the OR with a specific procurement protocol, so we usually have less than 10 to 15 minutes warm ischemia time, but we still see those. So living donor biopsies, these are usually obtained either before the kidney is explanted or at the backbench by the transplant surgeons in collaborations with our team. They get closer to the ideal state. And then there is an NIH workshop actually taking place next month on healthy tissue donors for research. And that really is happening. Collaborators of ours have actually from half a dozen of volunteers obtained kidney biopsy, which is a non-invasive procedure with minimal but present risk. And those form obviously an incredibly valuable reference tissue because you then can identify, even with small sample sizes, what are the principal differences under ideal conditions versus sample sizes where you get a more population level study, but obviously have the other constraints associated with it.
This see Christopher.
Hi, Christoph Cooper from Aachen. I got a question because we were talking about different compartments and how many data sets are actually available, like immune cells from the urine, because are there a lot of data sets available, and are they also linked to the kidney biopsy? So you can have basically 3 layers, like the PBMCs, the urine, and the kidney from the same patient. That would really link basically all of these compartments together.
Yeah. Also the AMP network for lupus nephritis had published 2019 the first paper where this was done. This was done with SmartSeq, so not with high numbers of cells. We actually have published a COVID paper with HCA support where we took urines from COVID patients and measured the intrarenal cell activation, including ACE2 receptor expression in people in multi-organ failure with AKI. And that study is expanded, and the data sets hopefully will be pushed out soon again. And multiple other groups are currently underway to try to do that. Procurements are under reference states a significant challenge, but with targeted QRT-PCR, so an ancient technology, but still effective, we were actually able to use cell type specific molecules and study those over the last 12 years and could show that podocyte loss, these filter cells loss, they have a very unique signature. And that signature in the urine predicts actually progression of disease, even treatment responses robustly. So there are evidence that there's a lot of information embedded, and finding innovative tools to get RNAs recovered out of the urine. As I know, together with Professor Graman, you are way on the way to do that. We're really looking forward to see that.
Great. So urine. I think we covered kidney quite well. We'll come back when we look at integrated at all the questions, but in order to have enough time for that, we're turning to Adipose. I'm going to do my best not to close your slides, which apparently I'm very good at doing. Margot is up next.
Thank you. Yeah, I have a very basic slide just going over the data sets that we're currently working with. As I mentioned yesterday, we have somewhat of an issue in the adipose field, which is that in order to get a full atlas of all of the cell types in the adipose tissue, we need to do single nuclear sequencing. This was very difficult to optimize because the lipids in adipose tissue make everything harder always. So there are only a few single nuclear data sets available. There are also some whole cell data sets available that we've been working with as well that are fairly complete except for the adipocytes. All of these that I have listed are currently published data sets. That's basically how we have been finding data sets, by seeing what has been published. And so that's definitely something going forward that we would like to do, is encourage people to share their unpublished data and to tell us that they have unpublished data that they could share, so that we could integrate that in. Another thing that I've listed on here are the adipose depots that these data sets represent. So I know that I'm speaking to mostly non-adipocyte people, so to briefly summarize, the fat depot, or fat is present in multiple depots throughout the body. The one that's going to always be the most represented in any data set, including this, is the subcutaneous fat, and that's because it's the easiest to obtain. It's right underneath the skin, and it's very easy to get from procedures like plastic surgery, breast reconstruction, basically any procedure that cuts somebody open, we could get discarded samples from the entry site. And so that's what most of the data currently available is. The second most available depot is the visceral depot. That's the fat, mostly the omental fat, that's in the peritoneal cavity, closer to organs like the liver and the stomach. This we can also get, especially from procedures like bariatric surgery and gynecologic procedures, actually, we've been getting a lot of samples from. Again, these are fairly easy to get as discarded fat from other surgeries, but less surgeries because you have to be in the peritoneal cavity. So one thing that we really want to do is expand the adipose depots that we're looking at. So a couple of the groups here have done that. One group has done deep neck brown fat, which is obtained using a needle biopsy, so that has to be something specific and not just a general surgery that was happening otherwise. And similarly, a group has done perivascular fat. I don't remember how they obtained it, but it definitely also has to be specially obtained, not just as a byproduct of a surgical procedure. And so we want to start collecting more of those, and that's really something going forward that we want to do. Things like perirenal fat, which are difficult to get without having somebody's kidney out, basically. Things like epicardial fat. We only have, an N of 1 of data sets of deep neck brown and perivascular fat. We want more of those fat. So that's something that the network is really interested in going forward. We also really want to get together and better define what the fat depots are because they're pretty big, and we're, taking random samples basically wherever the surgery is. And so when you say subcutaneous fat, most of us are actually talking about abdominal subcutaneous fat, but there's also hip and thigh subcutaneous fat, there's arm subcutaneous fat. And so we really, as a community, need to develop guidelines for how to define each fat depot and where we're sampling from, each fat depot in order to better, have metadata and understand our findings. So that's really the summary, I guess, of the Adipose network is that we're putting together some initial data sets, but we really want to get more data and we really want to get more depots. And that's something that I know my lab, Evan Rosen's lab, and Suzanne Mondrip's lab, the co-coordinators of the Adipose network, are working on. And we're really hoping to get more.
Before we turn to that, just a second before we turn to that, just for clarification for the general audience, and maybe coming, again, from my own ignorance, but I want to make sure this will be clear for people. So some fat, like the subcutaneous fat and so on, that is not associated with a particular organ. But there are organs, for example, the breasts, that it's kind of within the organ, and then there's like with the kidney, where it's like adjacent to, attached to, I don't know even what word to use, that organ. How much of that, if we had single nucleus or we had spatial profiling, two things where you don't dissociate cells, you don't put them inside droplets and wells, so all the problems of the cells are gone. How much of that do you expect to just be captured like immune cells get captured in stroma and vasculature and so on, and how much would require a dedicated?
So we definitely, in things like the skin and the breast, we definitely expect that to get captured naturally. And, I hadn't really thought of even it being captured in kidney samples because it's, as you said, on top of the kidney as opposed to within the kidney. But it makes sense that you would get some of those cells sometimes. Although I think that what we would want the most, and I know that this is getting into some of your questions, is an actual fat sample, either of the perirenal fat or whatever the sample is, because we aren't only interested in the adipocytes, we are also very interested in the other cell types, including the immune cells and the vascular cells. And it would be difficult to, I think, impossible to separate, what are the kidney vascular cells and what are the fat vascular cells. Maybe it would be easier than I think. I haven't tried. But we are really interested in the other cell types within the adipose depot as well, and the stromal cells also. But that said, I think that the adipocytes in other organs are also very interesting and something that we would like to either include in the atlas or use the atlas that we've developed using
To annotate.
to annotate because they, as you said, they do appear in other tissues, and that's
We have
also
seen
something.
them.
Yes, exactly.
Yeah.
Yeah.
And
So-
the main reason we have seen them is that we've done a lot of single nucleus analysis, so they
Right.
just pop up. We had them in the muscularis, when we did the colon muscularis, we had them in the muscularis of the esophagus, and we had them in the breast. These are the three where they, and the skin. As you would expect them to be there, but they're kind of like, here's some adipocytes, you don't really know.
Right, exactly. So yeah, we're excited that more people are adopting the single nuclear sequencing, because that's going to get us a lot of, as you said, just adipocytes from other organs that we can look at and compare.
Now
So we have a question from Giorgio in the University of Milano. So is there adipose tissue in bone marrow? And if so, he says that there should be plenty of it in the immune network.
That is a good point. There are definitely adipocytes in bone marrow, and there has been a lot of work done on bone marrow fat and on bone marrow fat progenitors. And actually, there has been, I know there was a mouse paper that specifically looked at adipose progenitors in the bone marrow. As far as I know, I don't know of an immune single nuclear data set that includes adipocytes from the bone marrow. But that would be great if somebody does single nucleus on the bone marrow.
Why it's not, because single nucleus RNA-seq is not the best method for immune cells.
Yeah, no, that makes sense.
And since single cells work so well for them, then of course, that's where people went in practice. But it doesn't mean that one should not do it as a dedicated effort.
Yes, we started with whole cell too, and had to change, but it definitely makes sense why the immune people would not.
Well, here's a data gap that you can leverage, right? Because I think every tissue can be sampled a little differently, and you can get other type of information. So for a bio network, actually, that would be very relevant, and they could coordinate with the immune bio network, so we could figure out interactions and so forth.
Yeah, fantastic.
Can I just comment? This is from Giorgio. So I'm trying to be Giorgio today. He says that healthy volunteers should provide the samples. Any comment to that comment?
Just in general? Yes. I think that's also something that it gets a little tricky when it comes to fat. Like I said, we are getting our fat at this point entirely, I think, from living donors, and so that is great. All of them are having surgery for some reason or another. That is less great. But generally, the surgery is unrelated to the fat. Like I said, like my lab specifically has gotten a lot of like gynecological oncology samples, and so we haven't seen any metastasis of, say, ovarian cancer into the fat. But if a sample were to have that, we would exclude that, because that would be too confusing. But in general, like what counts as healthy with fat, especially, we've been looking at a range of BMI, and there's a lot more to be done than what we have currently in terms of metabolically profiling the individuals that go into our data set. But you can't just set a BMI cutoff and say above this is healthy and below this is not healthy, because metabolic health, is a lot of different parameters, including things like glucose and insulin levels, et cetera. And so we have been doing things like excluding people that are on specific drugs that we think are that could affect the adipose tissue in ways that at least we would want to hold off and study later on. But yeah, I think the issue of health as it relates to adipose tissue is complicated.
Fantastic. Are you ready for my long series of
Yes, I'm ready.
I was given a long list of questions to ask people. I was asked, would you moderate this? Then handed the list. I'm joking, but it's a good list. Is there sufficient genetic, gender, geographic, and age diversity in the data sets that you kind of have in hand today? Are the metadata sufficient for diversity analysis? If not, what steps can be taken to increase it?
No. I feel like this answer session is going to go similarly to the immune one. All of the groups thus far that have contributed or that have data that's available at all are either American or European groups. Many of them are, I think, mostly white people. Actually, some of them, I don't know that we have all of the metadata to even tell that much. So yeah, that's definitely something that we as a network need to work on. We need to work on getting collaborators who are in other areas of the world, and we need to work on within our own areas, expanding our subject population. So that's definitely going to be a work in progress.
Cool. And now, actually, something occurred to me. Chloe earlier said, sorry, I should speak next to this. Chloe earlier said, and I know we have some people from the genetic diversity side. So Chloe earlier said that we can tell sex at birth actually from single-cell RNA-seq. She also participated in a study like that years ago. To some extent, we can likely tell ancestry from single-cell RNA-seq. It's genetic data. That is an interesting ethical question, because the data was not collected at the, usually sex would actually be collected and people just use it like a QC because you could have a technical error, but sex at birth is actually collected as metadata. Ethnicity sometimes and ancestry is not, but is actually recoverable from the data. So where is it, per sample, that is absolutely not allowed to do. You can't redo something that was not consented to. But in telling the overall diversity, say, of a data set, I wonder what our friends from both genetic diversity and if there's anyone from the ethics working group can share about that.
Well, from our experience, it's going to vary a lot from country to country because the approvals are different. Usually, if you are consenting for research, there are ethical matters in which you have to list everything that you're going to do. But depending on the approval, it's more general and it's for research. So all the information that can be collected from what you sequenced are okay. The most important thing is that it's anonymous, so you're not going to be able to refer back to the donor, so it's not an issue, but the donor has to know what kind of information you're going to be getting from it. Mm. But in principle, it will really depend on what kind of approval you have.
Great. Avi, there was this whole conversation in the good practice breakout session around this topic this morning. Because actually, it turns out that a lot of the groups, the consents they got, doesn't allow to kind of share that genetic information.
Yeah.
So the easiest way to share data is by, scrambling the genetic background, de-identifying the donors, which maybe is lesson learned for version 2. So we got to rethink a bit differently for this information. Yeah. Just some tissues you can, but there's.
Chloe, there's.
One of the things that we would like to discuss within this BioNetworks breakout sessions is what can be discussed before the groups collect the samples in order to make this more homogeneous and then maybe have data that we can share among all the BioNetworks.
Good lesson learned here.
Both on the consents and on the metadata.
I really want to stress that ethnicity, race, obviously for some ethnic groups are highly sensitive issues, and that even on a bulk analysis, as it is possible to deconvolve some of that information down to individuals, one has to be exceptionally careful not to provide information which is contrary to the beliefs of study participants. And this is really important. I brought it up earlier. One thing I'm really missing in our meetings is the participant voice. And obviously we have live people who in some instances put themselves at risk to donate tissue to research. And in most of our research networks, they are part of the leadership team, and they provide very valuable guidance to questions like the one we are just raising, so that we don't speak about people, but rather we let our participants tell us what they in their community would like to hear and see us doing.
I think that's a fantastic point, and we will absolutely make sure that we put it in the report back. Even though it was not in the particular context of any network, it's a general HCA point to make.
Matthias, I'm sorry I forgot to include this in our report back, but he did bring this up.
It will be in ours. I'm looking at
Thank
Jenny.
you.
So I put a lot of exclamation marks in my low-tech version of taking notes, as I am adaptable. Margot, I'm back to you.
Yes.
With my many questions. Are all relevant data identified / listed for integration? Is that it?
Yes, that's it.
Actually, it kind of seems simpler than Chloe's 100 million things in 1,000 papers.
Yeah, well, I mean, good at that. We wish it was a little more complex. I do know that there are some groups that are going to be publishing more single nuclear data sets, I think, relatively soon, and so we're hoping to get data from them as well. Yeah. But I don't know if we'll get it in under the line for the first draft.
The runner has a question. That's me. So you said you have this metadata. One thing you said earlier that picked up my attention is that, you said we randomly pick up fat wherever we can. And so as part of your metadata, do you track exactly where you took the fat from so that you can start building a framework? And how do you track it if you track it?
Well, so it's something that we need to do a lot better is the short answer. Most or everybody that's done this data thus far has given an overall indication of where it was from. For example, abdominal subcutaneous, leg subcutaneous, omental, et cetera. But one thing that we as a community really want to do is get together and work with some surgeons to better define, to really map out, I think similar to what the gut has really done in terms of finding landmarks and making a map that people can refer to when reporting going forward.
I think we share this landmark problem, immune and adipose, and probably like endothelial cells. Because we cannot come up with our own independently. We need to work with every tissue bionetwork to do so.
Yeah.
So I have to say, this is a great example, again, of an actionable cross-tissue integration project. Or a pre-integration project. If you ever want to integrate, you'd rather know where you came from. And when you work in a single organ, it's more about bringing the right anatomy knowledge, some of it being legacy knowledge. Another thing we will absolutely report back. But when you work in the distributed entire body, it is easy to kind of lose track. Yeah. So a great shared need. Yes. Chloe is not only the runner, she actually runs.
Yeah, she does.
To do it.
So I think, at least in the context of, say, obesity and all, we do see a lot of M1, M2, like macrophages and stuff. So, and there's always a question whether are they tissue resident or are they chemotaxing from other places. So in this context, like if you have paired samples, that would be one way to normalize what's, like what we were discussing in the morning, what's resident and what's chemotaxing. So in terms of that, is there data on these samples, on blood paired samples?
So is what you're asking, like, do we have like PBMCs that we can compare, like Chloe suggesting?
Contextual data, basically, because from what I understand, a lot of the fat cells have immune cells, like even on a 1-to-1 ratio, right? So how do you actually call it a fat unless if it's half of it is immune cells?
It's a really good question. In the currently available single cell data sets, I don't believe for most of them we have enough information there. There's one group, the O'Sullivan group that I've mentioned on this slide, who is more of an immune-focused laboratory, and I believe that they I think they might have flushed at least the samples before doing the sequencing, and I know that they did some sorting. There's, in general, in a non-single cell way, there has been a lot of research done on differentiating the tissue-resident macrophages versus the blood macrophages. And so I think that we can use some of that knowledge to kind of interpret our results, but we don't have a direct comparator like we've been talking about to specifically figure that out from the single cell data.
And maybe another question in terms of, when you said surgery, are there specific types of surgery? Because we used to get skin samples from bariatric surgery because they were just throwing it away. So we used to do immune cells with them. So are they like defined on what these patients or subjects are going to? Is it harmonized, or is it just random?
I believe that all of the samples listed here have metadata of what exact surgery they're undergoing. At least most of them do. But yeah, it's a good question, because obviously the type of surgery indicates whatever else is going on with the subject. So that's a good thing to have.
Thanks.
Great. I'm going to continue with my question.
Great.
Is the data available in raw as well as similarly processed format? And I'm asking along with it, is the data QC adequate and uniformly applied?
So for the first answer, no. Most of the data that we have is the process data matrices from whatever group collected the data. But that's definitely something that we can work on going forward. At the very least, like it was suggested in the breakout session, perhaps getting everybody to agree to some standardized pre-processing pipeline, at least in service of the atlas. Obviously, all of this data is already published, so they're not going to go back and change their paper. But that maybe is something that we could get people to do. In terms of quality, I think everybody has used fairly similar QC metrics. The metrics are going to vary between whole cell and single nucleus, both in terms of things like ambient RNA, which obviously is a lot more prevalent in the single nucleus, things like gene diversity are going to be different between the whole cell and the nucleus. So that's something that we need to, as a group, or at least we need a couple people to get together and decide on if we should, for example, apply the same cutoffs to all cells or different cutoffs for whole cell and nucleus.
The next one is one that I believe we answered through
Great.
our other questions. It's metadata. Do we have metadata on samples, protocols, sample location, preparation, sample location we just discussed?
Yeah, I mean, I think we have pretty good metadata. There's obviously some things that could be better. For example, more detailed information on where the samples are from. Like I said, I think we have all of the types of surgery, but I'm not sure. So we do have, basics like age and sex, but there are also things like metabolic parameters that are really not included in the data that we have for most of these data sets, that I really think going forward is going to be crucial for the interpretation of some of this data.
Great. How good is the current cell ontology for your system? And I'll add the second question right after. How different is individual annotation, meaning between data sets? Can we find a simple consensus, hierarchical, mapped to a cell ontology if not mapped already, and so on?
So I would say at like level 1, we're all in agreement. We all know what an adipocyte is. We all know what the pre-adipocytes are. We all know what the immune cells are as a whole. We definitely don't know what the immune subtypes are. And we have done some work to compare findings from data sets at kind of a level 2, level 3, especially in the pre-adipocytes and the adipose progenitor cells. So a number of groups have compared, mostly in the mouse data that's available, but also a little in the human data, the different pre-adipocyte slash adipose progenitor cell subtypes. And I would say overall they align well. They need to be called the same thing, and we need to get together and figure out what to actually call each of these populations, because every group has the name that they came up with, and then that's what they stick with every time they publish, even though another group is calling it something completely different. So, but I am really happy that at least it really seems like people are consistently identifying the same populations of some of these, especially pre-adipocyte subpopulations. So that's great. For other things like the immune cells, we really need to, I think, work on that. Again, there are some immune populations like lipid-laden macrophages that have been identified that people keep seeing. So that's also a good sign. But yeah, there needs to be, we really need to get together and agree on names, and and agree on some of the other subtypes as well.
Quick comment. It's just interesting thought, because I think both for kidney and for adipose tissue, you know, with your immune cells, it's coming from both cell and nuclei, right? And the integration of cell and nuclei will have the same resolution for defining immune cells, so that'll be an interesting integration challenge, Aviv. Cell and nuclei for defining immune cells across tissues.
Yeah.
The nuclei, yeah, that will be
Just like an interesting challenge to
It will be
kind
a
of
challenge
flag,
because nuclei cover better parenchymal and stroma, no doubt, we know now for sure. And cells enrich, they over-enrich compared to the actual tissue for immune cells, especially T cells,
yeah, it's just
which are underrepresented in nuclei. It's like, by now we know, yeah. No one's perfect.
something to think about for
Yep.
integration.
Very good point. I'll ask quickly, Margot, last two questions, and then we'll have a few minutes for things that are across the board, although we touched on a lot of them by now. I think I know the answer for G. Include all cells, how to deal with immune cells, stroma, etc. Include all cells,
definitely.
right? So that's an easy yes. Integrate all data sets or build an atlas using a curated subset and map the rest. You probably don't have the privilege yet.
Yeah, we are going to need all data sets in the end. In terms of things like, do we use references during the integration? It might make sense to use the single nuclear data sets as reference because they contain the adipocytes. Some of the, it could be, it could make sense to include samples that have all expected cell types as reference, and then in the process of integration to map everything else. There's also some differences, for example, between the subcutaneous and visceral fat. The visceral fat contains a mesothelial layer, whereas the subcutaneous does not. And so that would be really getting down to like one data set to use as reference, which I don't think is a very good idea at this point. But yeah, we do need to, I think in general for all of these data sets, we're going to really need to play around with trying different integration methods and see what we get, because I don't think that there's one straightforward way to integrate this data.
Great. Perturbations, you already said you have to take a range of BMI.
Yeah, like we're already doing a range of BMI, and we're definitely interested in doing things like specifically looking at diabetes. We also want to look at things like weight loss. And we also, like, even things like cold exposure, if we could get some people to, sit on an ice block for an hour
Just
or
move
something.
me back to Boston from the Bay Area.
Yeah, exactly. Yeah, well, there have actually been adipose studies just looking at people in different seasons and seeing the difference. So, right, so just Boston in the winter. But yeah, so there are definitely different states that we want to look at to include as part of the Adipose Atlas, because it will give us a better picture of what's going on.
Age?
Age, I think currently we have a fairly big age range. We don't have any pediatric at all, but we do have, I would say, ages in the 20s to the 70s. So we do have a pretty big age range already.
Very nice. Awesome. So we have like 5 minutes left, and I'm being generous. Yeah, we have 5 minutes left. I want to give us all a chance to reflect on three other questions. And they're big. I think we actually touched a lot on the cross tissue in many ways, doing it with PBMCs, capturing the fat when we go there, the analysis that can be done together, the finding coordinates together for immune. So I might set that one aside just because it came up naturally in the discussion. And I will ask the question, what are the current computational biology needs for this community in this room, for these bionetworks? And what are the three most important queries you would like to make on an integrated atlas, kind of in the hope of learning something important from it? Like, why are you bothering with all this hard work? Surely there's something you expect, a pot of gold at the end of the, that's I think the premise of the question. But also, what do you need computationally for your network?
I'll let you, I can certainly jump in, but please, go ahead.
I can say one thing that we've been talking about a lot also is figuring out some sort of website to kind of have information about the Adipose network on. Obviously, there are already a lot of really good options for actually uploading the single cell data or single nuclear data itself. But we really want to have more of an online presence so people know we exist and can contact us with their data. So that's something that we want to get set up. And, and we could put resources, protocols for doing the single nuclear sequencing, et cetera, on. That's one of our goals.
That's something that HCA can help with as well, because it has a well-attended website, and adding to a website.
Yeah, no, I mean, that would be great, yeah.
Yeah. Is that something that kidney and immune feel?
I mean, immune, we definitely need other than blood. Anything that's not blood,
Yeah.
that's an immune tissue. Because I think for all the other tissue, we'll rely on every other bionetwork.
Yeah.
And then once we see what other bionetworks have gathered, because we are aware not everyone is enriching for immune cells, we can figure out the data gap for version 2 if enrichment needs to be done for rare cells. I think for you're asking computational challenge, right, Aviv, and what are our needs? In the case of the immune system, it's not trivial, as I mentioned, because you have to think of it in terms of tiers across compartments, and then connecting compartments to each other. In addition to, it's not binary, it's a spectrum. And so we're oversimplifying our notation, and that's going to fire back. So these are a really hard computational challenge. In addition to, starting to think about integrating different data modalities and so there's no good existing methods yet so that we see the full facet of the cells emerge. In addition to understanding how they move between compartments. I could go on and on. There's a lot of challenges with immune cells. And there's obviously the annotation, but that's the first challenge we're going to try to tackle as a team. But I want to stress that for our annotation jamboree, we need our computational counterparts to be part of the conversation. We need our data generator, we need to be part of it. We need the immunologists and the domain experts to be part of it for this to be successful.
No, I just want to say, in immunology, it's really hard when you're required to put all your cells in little pots and call them by a type, because it just doesn't work. And I felt, we feel it when we hear what, say, Matthias and Gillian are saying about their kidney data. They say, but we need help with these immune cells, because they actually don't behave like yeah. Go ahead, Thomas.
I was just browsing the other network sessions online, and multiple of them said exactly the same thing. Like, we have these immune cells, and we have no idea what to do with them.
These immune cells are
We need
really
the immunologists to help, and it's quite good. Like multiple sessions that came up. So I think it's, I think that cross.
We should make sure we report that back.
Yeah.
I think
And that will be a great learning opportunity, because if you get...
And we actually want to work with all the bionetworks because we see no point in reinventing the wheel, so that we can all work together to figure out the data gaps and help each other with consensus annotation so it becomes more uniform. Yeah.
Matthias.
And then in the kidney, obviously, it's probably next to the brain, the anatomically and 3D most complex organ system, so we clearly need spatial data. And we clearly need multimodal data to see where the different transport and metabolic states of the organ is. They are target of effective therapies, so understanding the healthy state and how effective therapies are manipulating them is clearly one of the major needs of the community right now.
And I would agree for immune cells as well, because right now we're going to define some identity based on 1 or 2 modalities, and that's just a small facet of their identity. So as long as the community agreed that's not perfect, it's going to timestamp that annotation, and that annotation will likely be slightly wrong, and it's going to change over time as long as we agree on this.
Really attached to it.
Right? We just need to attach to how we defined it.
I think we're exactly at time. I want to thank everyone, but especially, first and foremost, Chloe, who took on many roles. Matthias and Gillian for the kidney, Margot for Adipose, and everyone for participating. Thank you very much. We'll be back in...
6 o'clock.
6 o'clock. We have 15 minutes to make all of this into one slide. One slide.